It is critical for quality and validation professionals to stay on the pulse of emerging policies and regulations. From the FDA's Computer Software Assurance Draft to the newly released EU Medical Device General Safety and Performance Guidance, evolving regulations are a constant. Our network of professionals cover these topics and more in print, in person, and online, bringing the latest industry news and tools to our audience of hardworking experts just like you. The IVT Network gives you the tools you need to succeed in your profession, providing innovative content, industry research, lifelong learning, and opportunities for networking on a global level. For our listeners, receive 15 months for the price of 12 plus an exclusive discount with your new subscription. Subscribe today with IVT Network, the best decision you'll make all year for your life sciences career.
This is Voices in Validation, brought to you by IVT Network. IVT Network is your expert source for life science regulatory knowledge. Voices in Validation brings you the best in validation and compliance topics. We interview industry experts from pharma, biotech, med devices, and laboratories. Here is the host of Voices in Validation, Stacey.
Thank you, and welcome back to another episode of Voices in Validation, brought to you by the IVT Network. This week, we're going to be talking about sources of contamination in pharmaceuticals and contamination control strategies. Contamination control encompasses all aspects of contamination, such as particulate, microbial, product carryover, chemicals such as cleaning material residues, as well as viral. And developing an effective contamination control strategy is important to the safety, patient safety, and of course, the integrity of the drug. So any strategy that may be adopted starts with a set of building blocks, but must be then adapted to the unique needs and activities of the facility. So here this week to discuss this process with us is Dr. Tim Sandle. It's great to have you back. Welcome, and thanks for being our guest once again.
Thank you. It's brilliant to be back.
Excellent. We always get so much, such a wealth of information from you. And to kick us off for this particular topic, I'm going to have you start off by telling us what the most common types of pharmaceutical contamination are.
Okay. That's it's fairly wide ranging.
Right.
So obviously we have microbial contamination, which would be the bacteria, the fungi, the toxic byproducts like endotoxin. Mm-hmm. Then there's also chemical contamination. So this could be things like residues left in vessels, where you get product compatibility issues. So if product flows down plastic pipework, you may get leachables or extractables.
Of course.
Then there's different forms of particles. And so these are particulates that could be classed as visible or subvisible. And these can be what's called extrinsic particles, which are things that get added, which might have come from, say, packaging material or fragments of a disinfectant wipe used in the process. Then there's intrinsic particles, which is where the formulation of the product goes slightly wrong. So you might get protein clumps and that kind of thing. Or there might be something relating to the active ingredient. Plus, there's also viruses, and there's also contamination that just results from everyday mix-ups and when things go wrong, things are not as clean as they should be. And so there's
so there's so many different sources, and you really have to, I guess you have to know what your process is and what the resulting issues might be. But what are the, you talked about the main sources and the different types of contamination. Are there any of these that are easier or more challenging to remediate?
I think anything involving people is always challenging because it's quite difficult to build in those procedural controls. But in terms of things that are easier, then I think with a new facility, it's always easier because the knowledge in the industry has progressed. So sure. clean room design, for example, is good, but it's still important to map the process first, because there's no point trying to fit a complex process in an already designed clean room. But legacy clean rooms obviously give a number of challenges. We've written on IVT before about the aging or the legacy facility.
Right.
So it's always good to start with process mapping and what goes on inside and outside the organization. Because even things like how items are transferred in can be challenging as well. Often heat maps can be useful as well, and the good use of risk assessment and staying up to date with current thinking. So, reading all the articles on the IVT network is a really good place to start for that.
Yes, absolutely. Thank you. Thank you for that. And so we'll talk a little bit more shortly about some of the strategies that you can employ to really help yourself along with this process. But I want to focus in a little bit more with a follow-up question on the chemical forms of contamination, because you did mention that as being one of the types of contamination. What are the chemical forms of contamination that are most common in pharmaceutical development and manufacturing?
Okay. It tends to be things like from moisture, from gases, from vapor, or even unwanted molecules, if we're getting into like biotech and so on, which can cause product adulteration. They can change the efficacy, or they can even cause sort of patient safety issues. But can also get contamination arising from the way the product's stored within the container as well, if the product starts reacting. And then also from cleaning validation. If cleaning validation hasn't been carried out properly, then there could be trace residues that are being transferred from one batch to another batch. And that's a recurrent FDA hot topic about, and particularly if there's any kind of manual cleaning involved, because it's not very reproducible. And all these things require good levels of detection for chemical, and often you need a combination approach. So it could be classic chemical analysis, it could be the use of spectrophotometric methods, elemental determination, even scanning electron microscopes, X-rays, and so on. So to make sure that you've got the required level of purity, and then making sure the drug is efficacious is really important.
Excellent. And so what does cross-contamination refer to then in this particular context? Is it, two chemicals, or is it two different forms of contamination? What would we expect?
Well, strictly speaking, cross-contamination is the physical movement or transfer of one contaminant in one place to a place where it's not wanted. So it could be via a person, it could be by an object, it could be by a piece of equipment, a vessel that's just been cleaned that gets parked too close to a dirty piece of equipment that's been cleaned down. You've got aerosols transferring over. Could be a person touching a surface and then inadvertently not spraying their hands and going somewhere else. Or it could result from damaged packaging. If packaging becomes damp or a container becomes cracked. So it's just the going from A to B when you don't want to go to B in this case.
Sure. Okay. So similar to what we would expect in a non-pharmaceutical environment, right? Yeah,
it could be kind of inadvertent where you get the general flow, or it could be down to poor procedures like mix-ups and adding the wrong ingredients as well.
Very good. So you mentioned that there's some hot topics or hot buttons for regulators. I'm going to ask you to expand on that a little bit and tell us what regulators, or from your experience, what do you think regulators are pushing for? Why are they pushing for a contamination control strategy right out of the gate? And what does that sort of look like under Annex 1 versus ISO or CFR?
Okay. Yeah, no, that's a good question. I think because obviously contamination control strategy is one of the kind of big buzz things around pharma at the moment. I think a lot of that's come from inspections, and it's the range of issues that have been picked up. And I think inspectors have seen examples of contamination. There's a rise in the number of recalls around the world, either due to chemical or microbial contamination. And then I think when inspectors then go into the facilities, they're seeing risk assessments that are substandard or have been written so they give the answer that the company wants rather than being based on sound science. So that's kind of the trigger point. Now, in terms of how contamination control features, it's kind of implicit in the CFRs and in the USP. But I think with Annex 1, it really kind of brings it out. Aside from Annex 1, there's the relatively new ISO 17141 standard, which is about biocontamination control. But that's quite specific. So Annex 1 is kind of asking a pharmaceutical company to consider all of their control measures and to bring all of those into kind of one document. So they're capturing design features, procedural features, technical aspects, organizational controls, and then the monitoring that's performed. And they're kind of centering this round quality by design, quality risk assessments, procedures, and how it all connects into the pharmaceutical quality system. So they're looking for evidence of all of that, all interconnected is the key thing. So often the word holistic's used, and that's quite a pertinent word to use in this context.
Right. So they are not only expecting you to maintain control, which of course we know is essential in pharmaceutical manufacturing, but they're really wanting you to plan for this well in advance and make sure you have your strategy and your protocols all laid out. So with that being said, in your experience, what does an effective contamination control strategy in the pharmaceutical manufacturing facility look like?
Yeah, I mean, it's going to vary a little bit between facilities, but there's a manufacturing element, a manufacturing control strategy. That's going to be based around different types of products and making sure those product lines are remaining pure and effective. There's also going to be variations in demand as well. There's going to be a quality control microbial aspect, which is about understanding the risk and controls, and that may be featured around critical quality attributes as well. And then there's going to be the cross-contamination control element as well, particularly when different types of products from different sources are taken into the facility. So there's going to be within that things like we mentioned about design of the plant and the process, personnel controls around the utilities and the cleaning and sanitization, raw materials, where they're purchased from, a bit about product design, product closures as well, and even things like vendor approval, where we're bringing in things like single-use disposable technologies. How have we selected that vendor? Where are we purchasing it from? How's that item been sterilized? How is it being presented into the facility? And then also things like outsourced activities as well. So there's a number of sort of disparate elements as well. We can add in process validation, process flows as well. So, you know, even with the development as well. So there's lots and lots of different things that need to be thrown into it. So the concept of the strategy becomes, you know, quite a thick book, but by the end of this. Absolutely.
I mean, yeah, sometimes we get all the way through the manufacturing process and we forget that, you know, the logistics pieces. So the transportation of the goods and the storage of the goods also need to be considered. And sometimes, you know, we fall down a little bit on those particular aspects of it. So I think having the strategy ahead of time from, you know, throughout the entire life cycle just helps us to prevent making some of these errors that could cause contamination. So, you know, as part of a contamination strategy, we often see a formal risk assessment. And that's important for identification and assessment of all contamination risks, as we were just talking about, from beginning to end. In your experience, should we be performing, you know, process simulations routinely or just for validation of the facility just at that time, for validation of the facility or the process, or are we doing this just on an as needed basis?
Okay, now that's important. And just on the risk assessment, the kind of key word is proactive. Risk assessment is best used when it's designed in advance rather than trying to put together something that's fallen apart. But with process validation in specifically, I think one of the key words that FDA has been very, very keen to emphasize is this whole idea of life cycle approach. So that's the principle that validation is not just this one-off activity and it gets filed on the shelf and gathers dust. It's about collecting real-time data as we go along and using that to keep on checking back to the original parameters, doing things like annual product reviews, setting in frequencies for revalidation and requalification. They're all important factors to consider. And when you get into sort of sterile manufacturing and you take something fundamental for process validation, like say media fills, for example, then they would need to be done at 6-month intervals because just the whole risk of that is too critical. With other parts of the process, then we can adopt a risk-based approach. But the key thing is about using the data and reacting to the data. And one of the great benefits, which we've discussed before, but is still kind of on the brink and is still to come, is the whole digital nature. We are soon going to have more and more technologies that can capture data, analyze data with big data analytics, and start to move to more kind of predictive analytics as well. So we're on the cusp of that, and that's quite an exciting area to be.
And it's going to help us be able to identify more quickly where things are going wrong. And to your point, probably in advance of when things are going wrong, because we can identify things in the system or in our process before an error actually occurs, which is so important and such a benefit of AI and digitization. So
absolutely.
Thank you. You were mentioning some indicators or, for some processes, like maybe looking at revalidation at 6 months or review of your process at 6 months.
Yeah.
But what are some of the key indicators or measures of an effective control strategy? And how often, ideally, should we be looking at these? And I get that with computerized systems, it can be more continuous. But if we're not yet there, what timeframes, what sort of benchmarks are we using?
Yeah, I mean, rolling backwards. I mean, the ultimate is not having any recalls, not having any adverse events, and passing FDA and European inspections. But within the actual process, I think it depends what we're trying to measure. I think things are a lot more advanced in the chemical world with rapid methods and assessing. It's quite easy to say, is this vessel clean by taking a conductivity reading, or with a circulating water system to have online total organic carbon measurement. With microbiology, one, you're often waiting for the result, and two, the methods are kind of more imprecise. But you can overcome that with lots of trending, lots of capturing all the different variables about knowing when the things happen. So, for example, if a process needs to be stopped and a part of the machine opens, you have greater confidence as to what's happening with that. But there are more technologies to capture data, and also I think having strong risk assessments as well. Knowing the weaker points in the process, knowing that if the path of a fluid slows down, then it reaches a certain level in terms of its turbulence or flow, then that might present a greater risk. Having data around how vessels mix and so on. So there's a lot of things, but it's building a big picture. It's amassing data. It's having as much automated as possible. It's also having about doing proper trending for sort of chemical and physical things. That a lot of that is statistical process control, and for microbiology, that might come down to the finer elements of QSIM charts and so on. But you've really just got to look at the data and be driven by it. And too many companies are just gathering data but not really analyzing it.
Yeah, that's fair. Unfortunately, sometimes they don't know what they should be looking for, right?
Yeah, well, the trouble is that a lot of people, somebody will come along, either go to an outside contractor and they set the system up, or someone would have worked at the company with a lot of head knowledge. They leave, and then it's like, what are we measuring? Why are we measuring it? What are we doing with it? I think it's getting better, but there's still some oddities within the systems.
So given that, what do you think some of the challenges are in developing and or implementing a contamination control strategy?
The biggest issue, if you don't have one, is time, because it takes an awful long time to gather everything and to prepare it and put everything together. And you can only really do that by involving all of your SMEs from within the organization, because you need the different subject matter experts. There's not one person who can sit down and write it. You're going to need chemists and microbiologists and engineers and process specialists and validation people all coming in. And then you may not you might end up with something very, very good with a few gaps, but you might end up with a strategy that's got a lot of gaps. And there's a lot of challenges, again, as we mentioned at the beginning, around legacy facilities. So you might have all the ideas for a fantastic contamination control strategy, but you can't actually implement it because the facility design just doesn't let you. And then you're into like, okay, well, do we then start risk assessing what we can't do, or do we try and seek investment? Is anyone willing to give us the level of investment? And then occasionally you can hit brick walls with some people who perhaps aren't as interested in delivering the outcomes as well. So there's a lot of complexities. So, if working in a medium to large pharmaceutical company, it could take a year to kind of get the strategy in place and written and down as a document.
So ideally, we have this control strategy. We've developed it before, but long ago, and we review it. Of course, that would be the right thing to do. But if they have need, let's say, for a new contamination control strategy because the one they had isn't effective, or they're just now getting to the point of putting pen to paper and really making this control strategy something that is written and people can refer back to. What are some examples of, contamination events that might be a catalyst for people to review or finally say, like, hey, listen, we need to, is it just that a regulator has identified something, or are there other examples of events which might be a catalyst, I guess, is where I'm going with this.
Yeah, no. A lot of things happen, unfortunately, and there's often once a month some kind of issue. So I'm trying to think of some recent ones that I've come across. So one was about endotoxin contamination in water systems. So this would come down to doing works by, say, changing a valve. So you have risks of cutting into the system, or the valve that's fitted may not be one of appropriate hygienic design, or perhaps the system isn't sanitized. It's good practice to, after the work, is to have the system flush into drain and not connected up to the rest. So that can be down to knowledge or specifications or quality of the work. Another area is the way that materials enter into a facility. So how well wrapped are they? Are they damp? Are they allowed to dry? Are they transferring? So a big thing with regulators is the concept of transfer disinfection. So going from a like a CNC area through the different grades of clean room, then they're expected to be like risk-based transfer disinfection steps in place as well, particularly going into, say, the place of final formulation, if it's non-sterile product, or the aseptic core if it's a sterile product. And then there's still quite a lot around cleaning validation. I think from IVT conferences and things, cleaning validation is a regular kind of hot topic. Yeah. And it's because it can be quite challenging, because pieces of pharmaceutical equipment are purchased and then it's suddenly found out they're not actually very easy to clean.
Right.
There may be little parts of the equipment that the cleaning fluids are not getting to. And we mentioned earlier about manual cleaning. It can be very variable between different operators, but even automated systems can be challenging as well if things get blocked or the blowdown procedure doesn't work and so on. So there's three examples. They're examples of real life contamination that everybody's facing.
And in your estimation, are some of those, they're great examples, and I can imagine that not all control strategies are created equal, right? There's going to be some procedures and processes that are better than others. So a lot of times when these failures or contamination happens, is it typically inadequate procedures or processes that led to inspection observations, let's say, by a regulator, maybe?
Yeah. I've got, so one example I could think of was the need to, again, going back to a water system. And this was a kind of a hot water system that was designed to like pasteurize a product. And it needed sanitization with a chlorine-based chemical. And it was working well for a long time because one experienced person was doing it. Then somebody else came along, and instead of doing a 1 in 10 dilution, they were doing a 1 in 100 dilution. So the efficacy of the sanitizer was
significantly different, right?
Yeah. So, and that was just down to a procedure that that the experienced person thought was really clear because they knew what they were doing. The inexperienced person couldn't quite follow it properly, so that didn't quite work out. Another one was I can think of was with pipe work that gets cleaned, and to protect it from cross-contamination, it was capped. But someone had an idea that to save money, would stop using these more expensive caps and would just tape paper bags on the end. But the paper bags were, if they got wet, then they ceased to be effective, so the pipe work could get contaminated. And then probably one more area that sometimes comes up, again, from procedures and inspections, is this concept of, often the phrase is used, like for like. Right. Which is okay if it really is like for like, but the problem is, like, a vessel, it becomes obsolete, a new vessel's purchased, and everyone says, oh, it's like for like, exactly the same, but it might mix at a slightly different speed, or it may not be as easy to clean, or it might not be the same surface finish. So we need to be very clear with our specifications, that if we are going to go down the like for like route, that we are still using a change control and we're still capturing everything adequately.
Absolutely. And in cases like these, some of the examples you just gave are great. I mean, they're real things that happen in the course of manufacture or drug production. Is training or retraining for the team typically the best solution? Or are there additional control measures needed to reduce the risks? And if so, how is that usually, I guess, accepted by the team?
Yeah, I mean, training can be, but I think it's often a regulatory criticism that training is sometimes used as the catch-all for everything. Yeah. And often it's often not the training, it's something more fundamentally wrong with say a procedure, or maybe there's a distraction. So examples there might be people who are tasked with inspecting product. Mm-hmm. So, how long are they going to inspect product for? What kind of light source have they got? are they likely to be interrupted? At what point will they start to get fatigue or eye strain? So there's factors around that, which isn't training. That's about job redesign and making the conditions better. Other cases might be with procedures, maybe using color coding in SOPs can help as well. Rather than just using black ink on white paper, you can use different colors or process flows and that sort of thing as well. And then also where we can introduce automation, then that's been proven to reduce errors as well.
Right.
And then occasionally making sure that the supervisor or the manager walks the shop floor as well, because if you go back many years, that was very common, but perhaps that's less common these days than it should be. So even that old-fashioned approach still has some kind of merit.
No, that's a great point. Just making sure sometimes people do the better work when they know that they're somebody might potentially be walking by, observing them, asking questions, etc. So and I think we forget about that a lot.
Sometimes
nowadays. Tim, how important is the environmental monitoring program to the overall contamination strategy in a facility? And I'm hoping that you can highlight special types of environmental monitoring and when they might be needed.
Yeah. So the first thing with environmental monitoring is really important, but it shouldn't be a substitute for poor control. So we always want to get like a good clean room design and good processes and people gowning correctly and cleaning and disinfecting. But you get that in place, then environmental monitoring can be very powerful, particularly when it's trended. However, it's very important to have undertaken a risk assessment to know what type of sample we're going to be using and what the location is as well. So, if we're in a big room and all of the personnel and the products kind of in that corner, we don't want to do the environmental monitoring over here. It's not going to be very realistic. So it needs to be kind of focused around the activity as well. So that's really important. And then also setting the appropriate frequencies of monitoring as well, and ensuring that we're adequately trending it. And then when we start analysing the data, we're looking at counts, but also incidences and recovery rates, and also speciating the microorganisms as well. So that's all important. And then in terms of sort of special types of monitoring, then there's areas like where we're using different gases. So if we're using compressed air or we're using nitrogen, then we're going to need to use different agars. We're going to have to use different atmospheres because we might be recovering anaerobes or microaerophilic bacteria that require special cultural conditions, for example. And we may want to apply that to also when we do things like freeze drying as well. If we're doing aseptic processing media fills, then we may need to do additional monitoring around that because we're kind of challenging the worst case. Depending on the types of disinfectants we're using, we need to make sure that the agars have the right neutralizers in there against the different disinfectants. And then depending upon the environmental monitoring kind of regime we're adopting and how we've qualified the media, we may need to do special monitoring for fungi as well, because that may not necessarily be recoverable on conventional media. So there's going to be different types. And also then for things like new facilities and startup for clean rooms, there's an expectation, particularly from FDA, that quite intensive monitoring's done to start with. So, we might assess a new clean room in operation for 10 consecutive days or more, maybe for a month. And then we can start to reduce that monitoring if we see the data looking better as well. So there's different kind of nuances and elements that need to be put in. Again, like anything like the contamination control strategy itself, the environmental monitoring should react to the changing circumstances, and it really needs to kind of be meaningful to what we're trying to do, which is showing, is there any chance of product contamination?
Right. And that makes total sense. That obviously you need to be flexible, right? And you need to know what you're trying to monitor and what your where your risks are, and all of that comes up front. So this is just a part of that, a continuing part of that whole contamination control strategy.
Yeah, I think it's always important to say, why am I doing this?
Right.
What's the purpose? And what am I going to do with the data? Because if you don't know those answers, then you're just gathering it for the sake of gathering it.
Exactly. Which is not effective. So, Tim, I'm going to ask you then to summarize for our listeners the key elements and activities in the biocontamination control for pharmaceuticals. Because we talked about a lot of different things today, so a summary would be helpful.
Yeah. So I think a biocontamination control in particular is about assessing the bioburden and microbial toxin controls in general. And then that might become more specific for non-steriles if we're focusing on specific types of organisms that might cause harm. And then with sterile manufacturing, we're trying to exclude all organisms from the process at all. And that's across all classified areas. And then linking into the contamination control strategy, what we're particularly concerned about is transfer or the transition of contamination. So it's often thought of that where you get the clean room, the different types of clean rooms like the ISO class 5, the class 7, the class 8, or the EU GMP ABCD grades, is very much like if you chop an onion in half and you look at it there, the center of that is the cleanest area. That's the aseptic core. And then you get the surrounding bits of the onion. And what you want to see is a reduction of contamination as you move through each one of those stages. So that's quite a good analogy for what we're trying to achieve with the overall contamination thing. And we need to make sure that it's holistic. So we need to be, if we're finding contamination in grade C or ISO class 8, we need to say, is there any chance of that getting into the ISO class 7 or the grade B? Do we have a risk with the transfer hatch, with the airlock, with the personnel changing rooms? We've got to try and connect up together what we're seeing to get the big picture. And then we need to say, where should we monitor either conventional methods or with new technologies, and what data can we gather in real time? But also one thing we haven't touched upon is having sound investigations as well. So, when things do happen, we need good root cause analysis. We need good investigational tools, and we need to be able to use risk assessment to say, what's the product or process impact? And then we need to be able to set good corrective and preventative action. So if the contamination control strategy is a living, breathing process, then learning the lessons and setting good CAPA and feeding them back in then helps with this culture of continuous improvement.
Sure. And that, of course, is essential to this whole process. And then hopefully, once you've learned from making some mistakes, eventually those happen less and less. And then I think the idea, eventually is to be as proactive as possible so you can avoid some of that reactive response.
Yeah. Proactive is the best place to be. Yeah.
Exactly. Fantastic. Well, thank you for summarizing that. And before we wrap up for today, I just want to see if there's any last thoughts or ideas, anything we didn't cover yet today that you'd like to leave our listeners with.
Yeah. So I think it's, the first thing is if you don't have a contamination strategy, then it's important to start putting the work in and design one. You might find that there are gaps, and it's important to acknowledge those gaps and not to hide them. You need to take them up to senior management and say, look, we've got to do something about these things. We might face regulatory problems, or we might end up with contamination where we don't want it. And then it's important to assess those gaps, to undertake risk assessments, and then start to extend the strategy to say, okay, when can we address the gaps? How long will it take? What do we do in the meantime? It's important to, one of the first things that we spoke about, that people, even with the best designed systems, can still sometimes do things wrong. So we always need to think about the human factors. I can write a procedure in an office, but what's it really going to happen when it goes onto the shop floor? Is it going to be treated in the same way? And then the final thing is that the contamination control strategy should not be seen as something static. It should be a dynamic document, and it should be regularly reviewed. And we owe it to patients to always be pushing for the for the best improvements that we can make to make things safer and reduce contamination events and things.
Absolutely. That's a perfect way to end this segment, Tim, because we always want to have patient safety as our overall goal.
Absolutely.
Well, thank you so much for sharing your information on this particular topic and being here with us today. It's always a pleasure to have you.
I'm happy to be here, and thank you very much.
Of course. Thank you again to Dr. Tim Sandle for being our guest today. It's so helpful to hear recounts of experiences in lab and learn from your knowledge on this topic. I also want to give a shout out to our producer, Ben Kitchen. Last but not least, a big thank you to you, our valued listeners. I appreciate your time each week, as well as your help in sharing this podcast. Let's keep spreading the word. As always, if you enjoyed today's episode, please remember to subscribe in your podcast player of choice and be sure to share it with your friends, colleagues, and online networks so they can enjoy it too. And lastly, for show notes and additional podcast resources, please visit www.ivtnetwork.com/podcast. We'll be back again next week with another noteworthy industry discussion. Until then, make it a great week.